Now, I want to take a moment here and think about some of the differences that we see in prokaryotic transcription versus eukaryotic transcription. In prokaryotic microbes, transcription is occurring in the cytoplasm. Remember, a prokaryotic cell, a bacterium, or an archaeal organism, does not have a nucleus. The nucleoid is located in the cytoplasm. So that means after that region of the gene gets unfolded, transcription is going to occur right there in the cytoplasm. It's very different in eukaryotes. Because of the nucleus, transcription in a eukaryotic cell is going to occur in the nucleus. The DNA will be unwound in the region of the gene, and the RNA polymerase will do its work right inside the nucleus. So in a eukaryotic cell, that means that the messenger RNA has to leave the nucleus and travel out into the cytoplasm, where it will be interacting with the ribosome for translation. In prokaryotic cells, there's no need for that because the mRNA is already in the cytoplasm. So in a prokaryotic cell, as soon as a short piece of messenger RNA has been produced, translation can start. It's different in a eukaryote. The whole messenger RNA has to be made first. And then that messenger RNA has to travel out through one of those nuclear pores out into the cytoplasm. And then translation can begin. So we describe it in eukaryotes as being sequential. First, transcription happens, and then translation happens. But in prokaryotes, the two things happen simultaneously. Translation will begin before transcription has ended. As soon as there's a little piece of messenger RNA ready for translation, the ribosomes will attach to it and they will start to translate it. So the DNA has the RNA being built on top of it, and the RNA is essentially growing off of the DNA, sort of hanging off of the DNA as it's being built. And the ribosomes will just attach themselves to that growing messenger RNA. The other thing to remember is the messenger RNA that gets made during transcription won't be translated one time. It will be translated again and again and again as long as that messenger RNA survives, as long as it's present in the cytoplasm. Now, I mentioned earlier that messenger RNA doesn't live long. It gets degraded pretty quickly. But as long as it's present, it will get translated multiple times. So that means from one messenger RNA, the cell will get multiple copies of that protein. Now, when you look at this under the microscope, you see a very unusual looking structure. You see this region of DNA, and you see some mRNA being produced from it, sort of hanging off of it, and you see all these ribosomes attached to the messenger RNA. Some people describe this as looking like beads on a string, and I'll show you what this looks like in just a minute. The technical term for this is polyribosomes, multiple copies of ribosomes attached to one messenger RNA. So on the bottom of this slide here, you can see an artist's drawing of this process of transcription and translation in a prokaryotic cell. They've drawn one strand of DNA here. Of course, there would be a second strand at the top of a bubble here. All the DNA back here would be in a double-stranded form, and all the DNA up here would be in a double-stranded form. But we've opened up the DNA in the region of a gene, and this would be the template strand. They're showing us this little gray structure traveling along the DNA. That's the RNA polymerase enzyme. Notice there's not one RNA polymerase enzyme here. They've drawn 1, 2, 3, 4 of them. Again, as long as this gene is exposed, as long as it's open, we're not going to make one transcript. We're not going to make one mRNA. We're going to make several mRNA. And then each one of those will be translated, not once, but several times. And that's what we see here. Here is an RNA polymerase coming in, and you can see it's made a stretch of mRNA that's about this long. Okay, so what's happening is ribosomes, and they're showing us those in this tannish brown color, ribosomes are attaching themselves to that mRNA, and they are translating it. So you end up with this string of mRNA with a bunch of ribosomes attached to it. And obviously, as the RNA polymerase travels down, and in this drawing, transcription is going this way, the string, the mRNA string, gets longer and longer as the RNA polymerase gets farther and farther down the gene. And as the string gets longer and longer, there are more and more ribosomes on it doing the work of translation. They've blown up this little ribosome right here, so we can see this little white chain here, the circles. That is the protein that's being made from this red mRNA. Polyribosomes, or beads on a string. Up at the top of the slide, you are looking at an actual image of this process going on in a real prokaryotic cell. This is an image that was made using an electron microscope. So we know from the central dogma that transcription occurs when DNA is used to make RNA. The next part of gene expression is this translation process, and we'll be switching gears here to talk about how that happens in our microbial cells. Remember, ribosomes are the machines of translation. Ribosomes are going to bind to the mRNA just before the start codon. In other words, they're going to come in and bind so that when they start moving, the very first codon that they will hit is the start codon. And remember, there's only one start codon, and that's AUG. Those are the 3 bases in the mRNA that the ribosome reads as being start here. So as the ribosome binds just before, just upstream, if you will, of that start codon, it's going to initiate this process of translation. And then the ribosome will physically travel down that length of mRNA, exposing one codon at a time. And a codon is simply a group of 3 bases in the messenger RNA. Each codon is read by the ribosome, and I have that word in quotations because what's essentially happening is the ribosome exposes the codon. It opens it up to tRNA, which are coming in and delivering amino acids. So whatever tRNA matches that codon, it will bring in an amino acid and give it to the ribosome. The ribosome will then move down to the next codon, and it will accept the next amino acid, and so on and so on. We refer to this process as elongation. amino acids arriving one after the other according to the code in the codons, and the ribosome binding those amino acids together to make a polypeptide or an immature protein. Finally, the ribosome is going to hit one of the stop codons. There are 3 of these. When it hits the stop codon, that will stop the process of translation, and we refer to that as termination. The ribosome will release from the messenger RNA, and that polypeptide, which it has built, that long strand of amino acids, will be processed and folded and turned into a functional protein. Now, the tRNA molecule has a critical job to do here. The tRNA is bringing the amino acid in. According to the codon, it's bringing in the amino acid that matches that codon. And it's giving the amino acid to the ribosome so that it can add it on to the growing chain of amino acids. Now, how does the tRNA know which amino acid to bring in? Well, first of all, there are individual tRNA for each amino acid. So there's a tRNA that carries the serine, and there's a tRNA that carries the methionine, and there's a tRNA that carries the phenylalanine, and so on and so on. There are 20 different amino acids. There are 20 different types of tRNA. On one end of the tRNA, the amino acid will be attached. At the other end of the tRNA, there are 3 bases in what we call an anticodon. So in order for a tRNA to give an amino acid to the ribosome, that tRNA has to have an anticodon that matches, in a complementary way, the codon that is being exposed at that moment. So the ribosome exposes a 3-base codon, and whichever tRNA has the anticodon that can bind to that in a complementary way, that tRNA will come in and it will donate its amino acid to the ribosome, and the ribosome will add that amino acid onto the growing chain of amino acids. Remember, the genetic code is universal among all living cells on the planet, microbial cells and other cells. The genetic code contains all of the codons that are found in mRNA. In fact, there are 64 of them. 64 different codons that code for the 20 different amino acids. And you might say to yourself, well, wait a minute, if there are only 20 amino acids, how come there aren't just 20 codons, 1 codon for each amino acid? And the answer is we have to remember that there's a little room for error in the genetic code, and that's a good thing. We refer to the genetic code as being degenerate. That's the technical term for this. What that word degenerate means is that there will be more than 1 codon per amino acid. There are at least 2, and sometimes there are 4 codons for an amino acid. And the reason I said that's good is because that allows for some mutation to occur without changing which amino acid gets placed where. Remember, it's important to a cell that its proteins be made correctly. And by correctly, I mean that it has the correct order of amino acids inside of it. So if a mutation occurs, it's in the cell's best interest if that mutation maybe could be a little bit degenerate in that the cell will look at it and still put the same amino acid there. Now this degenerate code has its limits. As it turns out, and we'll look at this in just a sec, it's the third base in each codon that has this room for error. The third position in the codon is the spot where mutations are best tolerated. In other words, for some amino acids, you can put any base in that third position. You could have a mutation that put any base in that third position, and the cell would still place the same amino acid at that location. And that's good. That helps a cell sort of weather certain mutations, sort of get through certain mutations without having it affect its proteins. Take a look at the code. Remember what this table shows us. It's every possible combination of the 4 bases that are found in mRNA. Every possible combination of A, U, G, and C in groups of 3. Okay? Now, what they're showing you in each box is the different combinations of 3 bases and which amino acid they code for. So, for example, in this box right here, THR stands for threonine. That's one of the 20 amino acids. So if the mRNA has a codon that reads ACU, the ribosome will place a threonine at that spot. Now, that third position where the U is that sort of wobbly spot, the spot where we can tolerate a little bit of mutation. Let's say that a mutation occurs in the DNA and it changes the mRNA to read ACA instead of ACU. Well, that's okay, because ACA in the codon will still place a threonine at that position. So that third base has the most resilience, if you will, to mutation. Now, not all amino acids are coded with 4 codons. Some amino acids only have 2 codons that code for them, and that's okay. It still gives us a little bit of wiggle room, just not as much as if you had 4 codons that coded for you as an amino acid. You can see the start codon right here, AUG. You can also see the stop codons up here. UAA is one of them, UAG, and the last one is UGA. I want to take one moment just to remind us about mutations. Remember, we said if there's a change in a base, we call that a point mutation. And we described different types of point mutations: substitution, silent, nonsense, and frameshift. When you hear the term substitution mutation, they're talking about one base being substituted for another in the DNA. That's going to change the order of the bases in the mRNA as well. And as we just said, depending on which base gets mutated, the codon that results may end up coding for the same amino acid, or it may not. So that's why we say that substitution mutations can, but don't always, change the codon enough so that we get a new amino acid at that location, and therefore we change the protein that's being made. Silent mutations are a type of substitution mutation. We see a substitution of one base for another, but the change in that base doesn't change the codon enough to change the amino acid that gets placed at that position. In other words, we call it silent because we wouldn't even know the mutation was there. It's silent. Yes, we changed the order of the bases in the DNA. And yes, that changed the order of the bases in the codon. But it didn't change which amino acid was placed where. That's a silent mutation. As long as all the amino acids are in the same place, you're going to get the exact same protein made. Nonsense mutations get their name because the substitution that occurs, one base for another, creates a stop codon where there didn't used to be one. In other words, you end up with a premature end to translation, a premature stopping point. And the protein is therefore much shorter than it should be, and it's not going to do the original job that it had. Frame shift is the strangest one because in frame shift mutations, it's not that we're substituting one base for the other. Instead, we're either adding in an extra base or taking out a base altogether. That's going to shift every single codon beyond that point. And that could change every single amino acid beyond that point and give you a very, very different protein.